Welcome to Biology at Priyojit, my dear viewers. Today we will discuss the various types of gene mutations and must understand how mutations are detected using classical genetic methods in Drosophila, that is a fruit fly. We will learn about point mutations, insertions, deletions, frameshift mutation, base substitution, and then focus on two important experimental techniques in Drosophila for detecting mutation, that is CLB method and the attached X method, which are usually used to detect X-linked recessive lethal and visible mutations. These methods helped scientists like H.J. Muller prove that mutations can be induced and precisely identified. This topic is extremely important for UG, PG, CESA-NET, GATE and other competitive exams. Let's explore how careful genetic analysis reveals hidden changes in DNA. So, what is mutation? Mutation is basically a sudden heritable change in the genetic material or DNA of an organism. The term mutation was firstly coined by the Dutch botanist Hugo de Vries. Occurrences of mutation, it can occur in any stage, in any cell, at any stage of development. Nature of mutations, it is usually a random process and it is generally non-adaptive. Result of mutation, it produces new alleles and it leads to novel traits called mutants. So what is the importance of mutations? It creates genetic variation. It acts as the raw material for evolution. It helps in identifying genetically controlled traits. So what is gene and allele concept? A gene is a specific sequence of nucleotides that usually codes for a polypeptide, and a mutation in a gene results in a new form, an alternative form of that particular gene is called an allele. Phenotypic effects of mutation. Mutations may show different effects on phenotype. Very small changes which are detectable only by special genetic or biochemical tests, visible changes that affect body structure or function, and severe effects which may cause death, that is lethal mutation. So there are types of mutant alleles, isoalleles, mutant alleles showing no visible effect and the effects are very slight and detectable only by special methods. So they are called as isoalleles. What are null alleles? Mutant alleles that produce no protein or produce a completely non-functional protein, those are called as null alleles. Recessive lethal mutations. If a null mutation affects an essential gene, homozygous individuals die, and such mutations are called as recessive lethal mutation. Dominant versus recessive mutation. Mutations can be either dominant or recessive. In haploid organisms like bacteria or viruses, all mutations are expressed because there is only one copy of each gene. In diploid organisms like human or fruit flies, recessive mutations are expressed only in homozygous condition and they remain hidden in heterozygous individuals. So lethal mutations are basically genetic mutations that lead to death, whether over a period of time or immediately after the mutation occurs. Now we study about the X-linked recessive mutations which are expressed in the heterogametic sex, males in humans and fruit flies and females in birds, because only one X chromosome is present. X-linked recessive lethal mutations, hemizygous individuals having only one X that carry the mutation die. This can alter the sex ratio of the offspring. General characteristics of most mutations studied: most are harmful or deleterious and most are recessive in nature. Conditional lethal mutation, they are most useful for genetic studies. They are lethal under one condition, that is restrictive, and survive under another, that is permissive. So it helps study essential genes even in haploid organisms. Mutations are grown under permissive conditions and tested under restrictive conditions. So it is used to understand development, metabolism, photosynthesis, etc. Types of conditional lethal mutants: auxotrophic mutants, they cannot synthesize an essential nutrient just like amino acid, vitamin, etc., and can grow only when the nutrient is supplied. Temperature sensitive mutants, that grow at one temperature but not another due to unstable proteins, enzymes inactive at non-permissive temperature. Suppressor sensitive mutants, they survive only if a suppressor gene is present and suppressor compensates for defective gene. Germinal and somatic mutation. This is the image showing the differences between germinal and somatic mutation. Germinal mutation occurs basically in germ cells and inherited by offspring. The dominant character, they appear in first generation, but recessive, they hidden in heterozygotes. Somatic mutations occur in body cells. They affect only the individual, not inherited. Heredity versus acquired mutations. Hereditary, they are inherited from parents and present in all cells, are acquired, arise during the lifetime, that is by means of radiation, chemicals and replication errors, not inherited. Functional types of mutations. Mutations basically are of various types, like missense mutation, where one amino acid is replaced by another, that is the image showing the missense mutation. Nonsense mutation, when codon becomes a stop codon, then it is called as nonsense mutation. Silent or neutral mutation, when no change in protein due to genetic code degeneracy, that is silent mutation. Gain of function, where new or enhanced protein activity is achieved, that is gain of function mutation. And loss of function, that is reduced or no protein activity, that is loss of function mutation. Conditional lethal mutations occur only when and only occurs only specific conditions. Forward and reverse mutations. Forward mutation, that is it is the conversion of wild type to mutant, and reverse mutation is the reversion, that is mutant is converted into wild type. Back mutation, when original DNA sequences are restored. And suppression mutation, when second mutation compensates for the first one. Spontaneous and induced mutation. Spontaneous mutations occur naturally, that is replication errors or unknown causes. Induced mutations are caused by mutagens like X-ray, UV chemicals, UVA chemicals, etc. H.J. Muller in 1927 proposed that X-ray induced mutations, and he achieved the Nobel Prize. Mutation rate, in case of prokaryotes, the mutation rate is 10 to the minus 8 to 10 to the minus 10 per nucleotide, and in case of eukaryotes the rate is 10 to the minus 7 to 10 to the minus 9 per nucleotide. So mutagens can raise mutation frequency to more than 1%. Point mutations, when single base changes in DNA and may alter amino acid and protein structure. Genetic code degeneracy provide partial protection of the point mutation. Frameshift mutation, when caused by insertion or deletion of bases, shifts the reading frame of codons and produces abnormal proteins and early stop codons. So the image showing here, frameshift mutations has been occurred by means of insertion. Insertion and deletion, it is caused by acridine dye such as proflavin, acridine orange, etc. It distorts DNA helix and it causes frameshift mutation. It helped prove triplet nature of genetic code, which was proposed by Crick and Brenner. Base substitution type transition, where purine to purine and pyrimidine to pyrimidine is converted, and it is caused by base analogs like 5-bromouracil, 2-aminopurine, nitrous acid, heat, etc. And transversion, when purine is converted into pyrimidine or pyrimidine is converted into purine, it caused by episides, and chemical like ethyl methane sulfonate causes transversion. Intergenic suppression, a second gene, usually tRNA, corrects mutation in another gene. This is called as intergenic suppression. It can suppress nonsense, missense, frameshift mutations, and suppressor tRNA reads faulty codons and restore translation. Mutator and anti-mutator genes. What do these genes do? These genes control how often mutations occur in DNA, and they mainly work by affecting the proofreading ability of DNA polymerase, the enzyme that copies the DNA. Mutated gene, DNA polymerase has poor proofreading, and in case of anti-mutated gene, DNA polymerase has very good proofreading. In case of mutated gene, more mistakes occur during DNA copying, but in case of anti-mutated gene, mistakes are quickly corrected. Mutated genes results high mutation rate, anti-mutated gene results very low mutation rate. So more errors is more mutations is found in mutated genes, but in case of anti-mutated genes there is better correction and fewer mutations. So example, fudge T4 DNA polymerase mutants, where some mutants act as mutators and some act as anti-mutators. So mutated genes increase mutation rate by reducing DNA polymerase proofreading, while anti-mutated gene decrease mutation rate by improving proofreading ability. Detection of mutations in Drosophila, that is by CLB method. CLB means curly lethal bar method. That is CLB is a simple technique to develop by Muller to detect recessive lethal mutations on the X chromosome of Drosophila. Basically, H.G. Muller was the first scientist to prove that mutations can be caused by external factor. He showed that X-rays can induce mutations in Drosophila. For this discovery, he received the Nobel Prize in Physiology or Medicine in 1946. So what are the components of the CLB chromosome? The CLB chromosome has three important parts. C means crossover suppressor, where a large inversion that prevents recombination, so mutations are not lost. I is the recessive lethal. The I means no, L is the recessive lethal, which causes death when present alone in males or in homozygous female. B is indicates barred eye, that produces narrow bar-shaped eye, making flies easy to identify. How did Muller perform this experiment? Male flies were exposed to X-ray to induce mutations within them. These males were crossed with CLB carrying females. Barred-eyed daughters were selected. They carried the irradiated X chromosome, and each bar-eyed daughter was crossed with a normal male. The offspring were examined. How were mutations detected? If no male offspring were produced, X-linked recessive, X-linked lethal mutation present, or when if male and female appeared in 2.1 ratio, there is no lethal mutation. So detection was easy. Just check whether males are present or absent. Attached X chromosome. What is attached X method? In this method, a special female fruit fly called an attached X female is used. In these females, both X chromosome are joined together and always move as a pair during egg formation. So the eggs produced are of two types: eggs with both X chromosome, XX, or eggs with no X chromosome. How does this help detect sex-linked mutations? An attached X female is crossed with a male carrying a suspected X-linked visible mutation. During fertilization, females get the attached X chromosome from the mother and males get the Y chromosome from the father and no X from the mother. Since males have only one X chromosome, any X-linked mutation is clearly expressed in them. How this mutation is identified? If the male offspring show the mutant trait, the mutation is X-linked, and when females may not show the mutation because they have two chromosomes. Why is this method is useful? It is very simple and clear, and it is best for detecting sex-linked visible mutations, and there is no complicated genetic analysis is needed. So as we conclude, remember that mutations are the fundamental source of genetic variation. Their detection help us understand how genes change and are inherited. From learning the different types of gene mutations such as insertions, deletions and substitutions, to understanding classical detection technique like CLB method and the attached X method in Drosophila, we see how chromosome behavior and visible markers allow precise in identification of X-linked lethal and visible mutations. These pioneering methods not only establish the experimental basis of mutation research but also remain highly important for UG, PG, CSR, NET, GATE and other competitive exams, holding us appreciate how careful genetic analysis reveals hidden changes that drive evolution and disease. Thank you.